After data integration, we can do clustering, and there are different approaches. Can be density-based or graph-based, which we have a Leiden or Leiden algorithm, can be accompanies and hierarchical. And in general, clustering is an unsupervised machine learning problem. And what we are trying to do in this stage is to label clusters by minimizing the intra-cluster distance in a reduced expression space. And for this part of the data analysis, we are going to use Leiden algorithm. If you're interested to learn more, and then there is this link which I have left in the Jupyter notebook, which discuss in details about the differences between the Leiden and Leiden cluster. Typically, graph-based clustering performed in 3 steps. First, we build a k-nearest neighbor graph based on PCA radius data, which we have already done that when we were running UMAP. The optional stage is to construct a shared nearest neighbor graph, and the aim of this will be to strengthen the connection between cells. Last but not least is the detection of community or clusters by optimizing modularity, and the modularity are resolutions that we use in the function. These 2 algorithms, Leiden and Leiden, have already been implemented in the ScanPy under these 2 functions that we can use. And note 2 important points. One is that Leiden algorithm is no longer maintained, and for this part of analysis we are going to use Harmony integrated data. First, we load the data. And then we run the Leiden function. The default resolution is 1, and we can also specify other resolutions, and then we label them accordingly. So let's run it, and then we can also plot them in UMAP. We pass the coloring criteria to be these labels that we have in our object. And there we go. So in principle, the lower resolution that you use gives you fewer number of clusters. But surprisingly, this is first time I see this, that 0.4 have one more cluster. And there is no typo here. But anyway, that's interesting. It was new to me. And then with the 1 as well. But that's the general rule that I just told you. But anyway, so with higher resolution, you see that there is a tendency to split the data even more. Which one is correct version? That's the typical question in single cell RNA-seq data analysis, because we need to go forth and back many times and correlate it with biological knowledge that we have. So don't just select the one that looks more appealing to your eyes. We can also generate the dendrogram, and here is showing you the correlation or how close these clusters are in relation to each other. We can also generate a dot plot by specifying given genes. So this is the list of genes of interest, and then we use dot plot function. We pass the onData object, the gene, and also we specify based on which resolution to be grouped. And then we also set on the dendrogram. So there we go. Here we have the genes, and here we have the clusters, which have been reorganized based on their dendrogram and similarities. The size of the circles shows the fraction of the cells in a group, and then the coloring shows the average expression. For example, CST3, we see that the larger fraction of the cells show high expression on average in these 3 clusters. I'm not gonna go into details about chimenes and Leiden and hierarchical cluster. I skipped them. They are already in the Jupyter notebook, so you can run it as you want to make a comparison. But for here, for this we can generate some of the information. For example, we can plot the some of the metadata. So the way that we do it is that we just to show you how we are generating it. So we create a crosstab. We select two columns, Leiden 0.6 and type, which corresponds to status of the COVID and control. Just to show you how it looks like, the table. Yes, here we have the Leiden clustering as indices, and then we have the COVID and control, the fraction of the cells corresponding to these two states or type, and then we can visualize them by plot bar. This is exactly the same thing that I showed you here, and here we are plotting. This is the first plot. The second plot, we create a new crosstab using Leiden and also the sample. So let's look at these two plots. So here it shows the distribution. For example, sample cluster 9 shows the larger fraction of the cells to be from COVID samples, and 11 seem to be only control, which looks a bit suspicious, but let's see whether we can figure out in next QC metrics visualization. And then we have Leiden's clusters and x-axis and the fraction on the y-axis corresponding to the source of the samples. So for example, cluster 11, we have cluster control 13, control 14, and control 19. Only these three samples. This isn't a huge bias in terms of samples that we see. For example, one cluster corresponds to only one sample. That's not the case. But let's generate some additional plots. For example, now we are just swapping this plot. We want to have an x-axis samples and see how many cells are distributed in given clusters. There we go. We have the samples and we have the clusters colored based on the labels. So, for example, in COVID-17, we see that majority of the cells are located in cluster 0. We can also generate the QC matrix, present them as well in plot. So there we go, we have the clusters, and then on y-axis we have the QC matrix. So cluster 10 and 11 might be worthwhile to discuss a bit about them. So they have a tendency to have higher values in general for these three plots, especially for gene by count. And the only thing which can be important to pay attention is that a small fraction of the cells seem to be in these two clusters. So these clusters should be very small, but if we look at the UMAP. And it's more or less the same behavior when it comes to total counts. And for ribosomal and mitochondria, it doesn't seem to be super biased distribution. If you want to just pick another example is cluster 9, which tend to have lower counts of ribosomal genes. So that was this part for clustering.